So good morning, everyone. Welcome to today's program, which is being organized under the theme of World Environment Day. This program aims to spread awareness of requirement of urgent needs to reduce the greenhouse emissions and restore our ecosystem. The program also aims to engage the community at individual and institutional level to reduce the carbon footprint through our day-to-day small efforts. To begin the program, I would like to invite the dignitaries on the dais, Dr. Radaha Rangarajan, Dr. Venkata Mohan, and Dr. Ajit Shasany. And Engineer Kamal Jain, please. And Ranveer, yeah. Ranveer? So to begin the program, I would like to request Dr. Radaha Rangarajan to present a bouquet of flowers on the behalf of CDRI family. I request ma'am to address the audience for this program.
Distinguished guests, faculty, my dear students and participants online. A warm welcome to all of you as we commemorate World Environment Day. A special welcome to Dr. Venkata Mohan, Director, CSIR, National Environmental Engineering Research Institute, NEERI for short, and Dr. Ajit Shasany, Director, CSIR, National Botanical Research Institute. Thank you for joining us today. Today, our program takes a slight departure from our usual science and research-focused events to mark World Environment Day. The goal is to bring awareness to environmental issues and to encourage action to protect the environment. Why? The answer is obvious. We need to protect the planet for future generations. We need to see ourselves as custodians of the planet, securing clean air and water for us and for the generations to follow. To ensure these basics, we need to take steps today to change our approach to environmental resources. Whether we develop drugs or work in other areas of science, we can no longer separate ourselves from the environmental issues because the environment affects all of us. We are in the 6th month of 2025, and it has already been marked with plenty of climate chaos. It has been another hot year, but the record-breaking temperatures have not been during the summer months. People in India experienced the hottest January since 1958 this year. By February, the cool weather started to recede, and it was an almost immediate transition to summer, leaving with us a very, very short or non-existent spring. It was the hottest February recorded since 1900, 1.35 degrees above normal. The IMD issued its first heat wave alert in April, but May was cooler than usual. The monsoon made an early entry with rains in different parts of the southern part of India. Mumbai broke a 100-year record for rainfall in May, and the downpour severely affected several parts of the city due to the intensity and sheer volume in a short spell. Perhaps none of us need to be reminded about these climate disasters and the gloom and doom that inevitably follows such discussions. So I'm not going to take you down that path. Instead, let us think constructively about environmental stability and sustainability. If we ask ourselves, what is the key factor that is driving environmental change? It is greenhouse gas emissions. Greenhouse gases such as carbon dioxide, methane, nitrous oxide, etc. play a key role in heat trapping, leading to changes in weather patterns and climate disasters. Greenhouse gas emissions are generated in many ways, as Neeti just said, but most centrally in our usage of electricity derived by burning fossil fuels. Lowering our emissions, therefore, must be a prime target of our actions. Let us register our commitment to being an environmentally conscious campus and community and actively work towards reducing our greenhouse gas emissions. As we enter our 75th year and plot the course for our future R&D activities, let us fold in environmental sustainability into our thinking. To take a step in that direction, we are setting in motion 3 different initiatives today. We have just inaugurated a 1.2 megawatt rooftop solar energy plant, which will directly reduce our dependence on fossil fuel-derived electricity. We have also inaugurated a 500 kg bio-composter that takes our food waste from the canteen and the guest house and garden litter and converts it into manure. This reduces our emissions by reducing occupancy in landfills that ultimately release methane and by sequestering carbon in the soil. Finally, we are introducing CDRI monogram teacups to replace paper cups in which tea is served at the mini canteen, thus reducing waste generation. We hope that through these initiatives, our net carbon emissions are lowered. Of course, we have to plan for more, and we look forward to suggestions from the community. I want to thank the prime movers of these projects. Kamal Jain, Senior Superintending Engineer of the Electric Section, and his team. Ranveer Singh, Scientist in the Medicinal and Process Chemistry Division, and his team. And the Canteen Committee, headed by Dr. Rajkamal Tripathi, for spearheading the solar plant, biocomposter, and cup initiatives, respectively. To make this day very special, we have 2 distinguished guests with us, Dr. Ajit Shasany and Dr. Venkata Mohan. Dr. Shasany, Director NBRI, is a biotechnologist whose research interests lie in the areas of metabolic engineering of medicinal and aromatic plants, plant immunity to boost crop production, and plant biodiversity. Since becoming director, he's taken several steps to protecting the environment, including conducting studies of urban heat islands that are substantially hotter than other areas, and finding ways to mitigate such heat by planting the appropriate vegetation. Thank you for being with us today, Dr. Shasany. Dr. Venkata Mohan, Director NEERI, joins us from Nagpur. NEERI is an institute that is dedicated to addressing environmental challenges through scientific research and technology development. His interests are in circular bioeconomy, bioenergy, decarbonization, and waste valorization. He has also developed methodologies for wastewater surveillance to track antimicrobial resistance in the community. And he is a participant in the CSIR-AMR mission focused on drug discovery and surveillance. We are delighted to have you with us today. Before I hand the stage back to Neeti, I'd like to thank the 75th Year Celebration Committee chaired by Dr. Amir Nazir and co-chaired by Dr. Vinita Tripathi for their help and dedication and for making this event possible. And of course, thank you very much to all of you in the audience here and those online for joining us today. Thank you.
Thank you, Dr. Radha. Now we will play a video to give you a glimpse of the hard work and the concept behind the solarization of our campus. The Central Drug Research Institute is a constituent laboratory of the Council of Scientific and Industrial Research under the DSIR, Ministry of Science and Technology, Government of India. The institute is engaged in basic and translational research with a focus on drugs, diagnostics, and process technologies in disease areas of national relevance. To facilitate the research and development activities, our campus located in Lucknow has state-of-the-art facility equipped with a range of sophisticated instruments. Naturally, maintaining this infrastructure requires a significant amount of energy. Our consumption averages about 3 megawatt per month with some seasonal variation. This is not unusual for biomedical institutions, particularly those that are heavily engaged in experimental sciences. Most of India's electricity is produced by burning coal. This in turn generates carbon dioxide, a major greenhouse gas. Other human activities such as deforestation, industrial processes, and even our day-to-day activities like using electricity, traveling by car or flight, and consuming manufactured goods all contribute to carbon dioxide emissions. Sadly, these emissions have many adverse effects on our planet, which include rising global temperatures, melting of glaciers, destruction of ecosystem and biodiversity. To mitigate these effects, it is imperative to reduce greenhouse gas emission with efforts at individual, institutional, and national level. Transitioning to renewable sources of energy like solar and wind, improving energy efficiency, embracing sustainable consumption habits are some way to reduce the carbon footprint.
In 2024, Dr. N. Kalaiselvi, DG CSIR, flagged off an initiative together with Energy Swaraj Foundation to reduce CSIR's energy dependence. Embracing this goal with all seriousness, we brainstormed as to what we could do to make a significant difference to our carbon footprint. We emphasized individual commitment to reducing energy consumption, which remains an important priority. But at an institutional level, we explored ways to do more.
Rooftop solar energy generators have been the cornerstone of the Government of India's policy for environmental sustainability and energy self-reliance. Central to this effort is the grid-connected rooftop solar program by the Ministry of New and Renewable Energy, which incentivizes institutions, households, and commercial entities to install solar panels on available rooftop spaces. These systems are integrated with the local electrical grid, offsetting the cost of electricity proportionate to what is produced, thereby reducing the overall carbon footprint. Various schemes are available to promote and make possible the solarization of institutions.
The Renewable Energy Service Company RESCO model offers electricity to consumers from solar power plants without requiring any upfront investment from them. In this model, The RESCO company invests in and manages the solar power plant, which includes installation, maintenance, and repairs for a defined period, after which the plant is handed over to the consumer. Consumers pay for the electricity generated by the plant, usually at a predetermined monthly rate. Solar panels at CSIR-CDRI now cover 100% of the rooftops of the R&D building. In phase 2, we will aim to cover 100% of the rooftops across residences and administrative buildings. Our current demand for electricity is around 3 megawatt, whereas the peak generation by the solar plant will be around 1 megawatt. Factoring in the shorter winter days and occasional inclement weather, our rooftop solar plant will meet close to 30% of our requirement.
Biomedical research facilities need to plan for the future. Research and development activities must go hand in hand with environmental sustainability. We see this as our responsibility, and today we're taking a substantial step in that direction.
Let's put concerted efforts to harness renewable energy for a sustainable future. So you can imagine a lot of hard work has been gone and in solarization of the R&D block. And as Mr. Jain has said that in the phase 2, we are going to have solarization of the residential complex as well. So moving forward to another step towards building a sustainable environment, we have established a on-campus bio-composter, and I request Ranveer to please tell the audience about it.
Good morning to esteemed chief guest of today's program, Director NEERI, Director NBRI, Director CDRI, ma'am, and dear colleagues and friends and students. So before going in the detail of biocompost machine, I wish to seek your kind attention that CDRI moved from Chhatar Manjil campus to this campus, which was dedicated to nation by Honorable Prime Minister Sri Narendra Modi ji. Since then, our campus has prioritized green innovation and eco-campus initiatives. In line of this, we have 20 kiloliter per day capacity effluent treatment plant dedicated for the waste which is generated from laboratories and animal research facilities. We have 375 kiloliter per day capacity sewage treatment plant, and I am happy to share with you that all the greenery which you see in the campus, that is using the STP treated water from treated by us. We have separate dedicated garden lines to reuse the water in horticulture works, and also the CDRI campus, this campus is complied with rainwater harvesting provisions also. So today, with the inspirational and motivational leadership of Director CDRI, we have added one more step toward the green campus. We have installed 500 kg per day capacity biocompost plant on the occasion of World Environment Day celebrations. And this plant will utilize all kitchen waste, leaves, grass and horticulture unused wastes. This biocomposter technology is based on thermophilic treatment under aerobic conditions and homogeneous mixing, which converts the material to nutrient-rich bioorganic compost within 24 to 48 hour cycle. So as a process engineer point of view, the complexity of any plant which we measure in terms of number of unit operations and unit processes which are involved. So that information we extract it out through the process flow diagram, and also followed by front end engineering design, that in short form or in engineering terminology we call that feed data. So which provide the details of mass, energy and material balance information, and on the basis of this we decide the life cycle, process feasibility, and through which the data optimization of process parameter we can do, and this helps to reduce the recurring costs. So in terms of these requirements, the present technology and working of biocompost machine is very simple. We have choose. So waste material first goes to size reduction units, followed by the digester through the hopper, and the digester temperature we are keeping in the range of 60 to 70 degrees centigrade, and the continuous mixing with the rotation of agitator with the 10 to 12 rotation per minute. And rate of aeration improves the digestions and remove the odor generation during decomposition. So the decomposed product with proper aeration is free from moisture and odor and more than 80% of volume reductions. So 500 kg we are getting around 100 kg of the product from this. And this installation of machine is also in line with municipal solid waste management compliances and toward the achieving goal of zero discharge campus. And at the end of this, I express my sincere thanks to Director CDRI and guests at the dais to encourage and make the movement memorable to me and entire CDRI family. I express my thanks to my colleague Mr. Sivram Misra, Senior Technical Officer of my division, and ESD engineers Kamal Jain, Mohit Suklaji, Brahma Singh ji, Siddhu Hembram, Jay Prakash for their valuable civil and electrical inputs in day-to-day operation and management of biocompost plants. Thank you.
And as Director mentioned that we had 3 initiatives which we are launching for a sustainable environment efforts. The third one is the unveiling of or the launch of Bring Your Cup campaign. As you all may agree that CDRI chai is very, very good, but the downside is that we generate a lot of paper cups waste. And to reduce the carbon footprint, this is an individual effort that you all and we all can make as a community and as an institution in reducing the carbon waste and greenhouse emission is by using reusable cups. So I request the guests So we have one planet and we need to be part of that planet by making good efforts for a sustainable future for us as well as for upcoming generations. And this would be distributed to everyone in CDRI. So next time you stand at mini cafeteria, you have to bring your own cup.
And the tea will cost more otherwise.
Yes. Otherwise, you have to pay 3 times more. Okay. So now we move forward to the keynote lecture by our esteemed guest, Professor Venkata Mohan. Director has already introduced him, but I would also like to tell few things to the audience. Dr. Mohan is actually an engineer through formal training. He did BTech in civil engineering, MTech in environmental engineering, followed by doctoral work. He had been serving as chief scientist at CSIR-IICT since 1998, and in January 2025 he was appointed as director of CSIR-NEERI. His work has been recognized both nationally and internationally. He's recipient of Shanti Swarup Bhatnagar Prize, Tata Innovation Fellowship, Humboldt Fellowship, and he's also a visiting professor at Kyoto University. His research focus has helped in contribution in advancement of technologies in bioenergy, decarbonization, biorefineries, and his research work has led to many important patents and technology transfers. And today he is going to talk about surveillance and management of antibiotic resistance through wastewater pathways. And this is very important, because when we talk about One Health concept, so it is not only good health for us, but also animals, as well as the water health, as well as soil health. So we look forward to your talk, Dr. Mohan.
Dr. Ajit, Director, CBRI, Dr. Rangarajan, Director, CDRI, colleagues from CDRI, students, and distinguished guests. It's my honor today to stand before you to speak something on sustainability, which also has significant role on the eve of World Environmental Day. Before going into the presentation, I would like to say a few things that what happened today under the able leadership of Director CDRI, particularly with reference to sustainability. The three initiatives which has been taken today are very important in reducing greenhouse gas emissions, particularly the solar energy harvesting system that directly contributes to 30% reduction of greenhouse gas emissions, as it is contributing to 30% of energy requirements. Basically, our energy we source from fossil based resources. Our energy mix consists of 80% of fossil, 20% of renewable. It's not precise, but 80% it's say by record. So by way of implementing this, we are able to reduce 30% of the 80% fossil fuels emissions that is contributing to the environment. That is the first thing. And second thing, composting is one of the very sustainable process. The carbon footprints, particularly when we look into the carbon farming concept which is coming up. So we know that carbon content is reducing in soil. The fertility of soil is getting reduced. So these kind of composting practices, particularly in a circular way, particularly in campus, are trying to take the waste and trying to make into compost and use it again for a purpose that will have significant impact on reducing the carbon emissions as well as on increasing the sustainability. And apart from that, the waste water which is being generated in campus, apart from rainwater harvesting, that's one of the important initiative where few campuses are doing that, trying to reuse in the campus itself. These three will contribute significantly to reducing greenhouse gas emissions, also to address the environmental sustainability aspects. So I would like to congratulate Madam as well as CDRI for taking up of these steps, and also contributing to the sustainability on the whole. So coming to today's talk, Madam told me to, when advised me to take this concept, because CDRI is not my domain of expertise, but Madam told to talk about surveillance, particularly related to AMR, which is close to my area, which I am last 5 years doing since COVID. So I would like to, even though I am not a biologist, I am a civil engineer, but I have expertise in applying biology to environmental aspects. So I'm not a core biologist, so you have to excuse me if you are going deep into biological things, but application I'm strong, I would like to say. So going into starting with, I came from NEERI. Everybody know about NEERI. NEERI is one of the 37 among 37 CSR labs where it contributes significantly on basic applied research pertaining to environment, all domains of environment. Started in 1958, initially with respect to the jaundice. During that time, it started as Central Public Health Research Institute and started working on reducing these diseases by developing strategies for water treatment as well as disinfection. And slowly, with changing of time, with new environmental issues, it also changes its research as well as other activities to other domains, starting from water, wastewater, solid waste, hazardous waste, environmental impact, and now sustainability. So it moved and it addressed the requirement of environment with respect to Indian context. Indian supports policy as well as it's one of the only referral lab in India, particularly for environment. So coming to CDRI Connect with respect to my research, I have two projects, one mentoring by madam on AMR that I will speak a little bit in the presentation. The second one, yesterday we have a final presentation happened on API project where CDRI also partner. So where here sustainability is one thing which I am looking into the project in the production of APIs, where we try to use life cycle analysis to understand the hotspots with reference to the chemicals or the process inputs developed from lab scale to pilot scale. So during developing stage itself, when we understand the issues, then we can address at the final stage. That is the basic objective. In that line, we are trying to develop a methodology based on ISO 14044. So shortly, we'll be also looking into the data from CDRA to analyze some of the process came up to the higher level. So these are the two things I directly have contact with CDRA, and in future we are also trying to collaborate. Madam and we discussed on many things on sustainability. I'm very happy to collaborate with CDRA from NEERI side. So as I talk now, I will this presentation I have made into three stages, three phases. I would like to say the first one, why we started wastewater-based epidemiology. That is way back during COVID period we started working on that, and then I moved to AMR. How we shifted and why we shifted to AMR based on the same methodology, wastewater-based epidemiology, and then also try to present some of the data recently which we collected on wastewater treatment systems, how the fate of AMRs will change during treatment process in three of the treatment systems. So first I would start with COVID, which we started way back in 2020 during COVID phase with collaboration with CCMB, with Dr. Rakesh Mishra. So the basic objective is to understand we are able to detect COVID virus in wastewater system. So basically, by this time everybody know that there is one particular paper which came in April 2020 that they are able to detect virus in wastewater system, because we know that before coming up of the symptoms, 14 days before, there will be this virus prevalence, and after we recover from the illness also 14 days, on total 30 days this virus will be there in the human system, and particularly in gastrointestinal system, and along with the stools, this virus will come into the environment, one of the route. So here to connect this one, wastewater is one of the domain which carry all these things, domestic wastewater coming from our daily activities. So with this basis we started working on, can we detect virus, that COVID virus from wastewater is our basic scope. So we started working with as I told, CCMB, because CCMB has all facilities for virus, and we have expertise in wastewater infrastructure analysis. So we choose Hyderabad. Hyderabad accounts for about 10 million people. Our objective is to look into sewage treatment plants. So when we collect sample from sewage treatment plant, inlet of sewage treatment plant, we can say the infection rate or immunity of the community. So we know the quantity of wastewater which is coming to sewage treatment plant, inlet of the sewage treatment plant. No need to collect samples everywhere. So we try to get permission initially from various sewage treatment plants and collected samples. And these are the some strategies that methodology we have used. So these are some kits available at that time. So these are the calculations. A few calculations are available. We try to use these calculations based on RNA, wastewater, and feces, based on feces. And we collected sample. Initially, collection of sample also, this is a peak epidemic. At that time, infection was very severe, so we tried to establish a methodology to collect sample also. So taking all precautions while collecting and transporting. So during transport also, we don't want to avoid any contamination to the sample collector. So we try to establish a methodology that is based on hypochlorite. And also we try to make standard graphs to understand gene copies based on the real COVID genes, genes which is available in CCM. So this is the primary data which we got initially. So this is the first, I can say that whatever methodology we have chosen, that has given results. So here all STP plants with a different capacity in Hyderabad, which covers about 60% of the population. We try to be able to understand the infection rate at that particular point of time based on E genes, N genes, ORFL1 genes. So here in these heat maps, you can see the variation of 3 genes. Particularly in inlet, there is significant concentration of these COVID with respect to 3 genes, but outlet of treatment plants, there is no presence of any virus. Particularly, you can see all the outlets, there is no present. That is one of the good thing, because treatment systems are working effectively. As you know, it's a virus, it's very sensitive. So with harsh conditions, what is happening in treatment plant, apart from disinfection, we didn't detected any virus in the treatment outlet of treatment plants. So then we also studied different time periods, also collected samples from household community where few hundred families are there in order to ascertain is it working out or not. So we observed presence of this virus, and now the methodology is important. When we have to collect sample, how to collect sample should be representative sample, otherwise we will not get the correct data of infection. So by this time we did get permission from the municipality, because by that time newspaper, lot of news came from that publication that the city is infected so much and institute has identified. So government stopped giving our samples. So then what we have done, we know the inlet point coming to the sewage treatment plant. So the converging point which enters into the sewage treatment plant, we identified, and all our studies we have done there. So this is the point, the converging point to sewage treatment plant. We try to collect sample throughout the day, so to understand infection rates or CT values, when we'll get more, how it is varying. By this a kind of composite samples. If there are 2 kinds of sampling, composite and grab. So composite means, it's not working. Okay. Yes. So you can do the time we started. Oh, that's the way. No, it's fine.
Now it's
yeah. So you can. We try to collect sample starting from 5 a.m. to next 7 a.m. Continuous to every hourly sample, and you can see the CT values here. We can observe RNA copies or CT values are higher with infection rate, particularly from 5 a.m. to 12 noon, about 9 a.m. So during which most of the domestic activities will be there. So then we also studied composite samples, and we observed that either going with sample collected during the grab sample by single sample between this period of time, or a composite sample throughout the day which we collected, will have more or less with 5% variation. So we try to standardize the methodology. You can see variation during different times during the collection of different time periods. So we come up with a methodology, and this also matches with the infection rate at the time reported in the Hyderabad. So this is first methodology workout we have done in these kind of studies where SOP also came. This SOP was also reported in WHO guidelines. So then we try to look into long-term surveillance. So getting sample 1 or 2 times, getting infection is not a correct thing to understand the community health. So we try to collect the samples from that area where you can see the map. We try to identify different drains which is emanating to the effluent sewage treatment plants. Near to 10 samples from different locations, from main drain to lateral drains, we try to collect, and we try to look into the infection rates. That in graph you can see RNA copies varying. Rainfall, when rainfall is there, we cannot collect sample because water get diluted. So during that time we will not collect sample. So there is a significant variation. This matches with the infection rates report by the state government. Next, please, madam. Yeah, yes, madam. Thank you. So then here interesting thing, we started working from July 2020 to 21st May 2021. Continuously sample frequency once in a day, once in a week, like that we have done. So the whole period you can see first wave where RNA copies dropped, particularly the infection dropped here, and suddenly in March you can see from February there is a rise which emanated for a second wave. So this is a very strong wave which we observed at that time. So but this is an early warning signal we got from these studies. But at that point of time we don't know that infection is getting increased. But once all this happened, when we plot the data, we observed we got signal early only. So initially we know that something is happened, we try to take some kind of precaution. This is the thing which we understand after the second wave. So this kind of data also we can get, and also community zoning. You might be knowing zoning based on the infection. That also we can do. This is the community which we have studied. So we collected all the lateral drains, the main drain, and based on the infection rate, we can zone the community which is having more infection, so that this will get some kind of closer kind of thing, so that infection will not go into the other areas. So this kind of zoning also we can do with this data. This has done during that COVID time. So these are some of the things that just want to explain. And here we also observed the lakes which is coming, taking the domestic sewage. These lakes in Hyderabad also showed similar kind of pattern compared to the domestic wastewater. So that is regarding that one. And this study also helps us to understand the prevalence of variants. So this during INSAC project we have done. Wherever we got good peaks, we try to understand what kind of variants are present. So the second stage of study with the support of DBT in INSAC project we try to detect the variants during almost 1 year of time. So these kind of things also we can do with this study. This is the recent sample which we collected, only one sample from Hyderabad, but we didn't see only RDFA gene, only one gene is able to detect an RDRF, but other E and N genes we are not able to detect. So coming to next thing, what AMR, antimicrobial resistant genes, we try to look this also in the way of wastewater based epidemiology, because we know now gut bacteria is coming in a big way. So the bacteria or stools which is coming in the domestic system will able to say antimicrobial resistant of a community. That's the basic thing, and we try to look into this one. This we are trying to work from 2021 November. Once we started working on COVID, we started working on that. So initially we took the same study area, urban community, and then we extended to other places like wastewater treatment and Mahakumbh also. We try to look into the antimicrobial resistance. So this is the methodology, and a very similar methodology, but a little bit analysis. We try to use culture independent target specific strategy. 123 antimicrobial resistances depends different class, and apart from 13 mobile genetic elements, we try to study during this one. And the sample collection we normally done, except rainy days, we have done between 7 to 8 a.m. with a grad sample, and a cold condition we transferred to the lab. So these are the data which we got initially. So with respect to the resistance with different class of antibiotics, antibiotic resistant genes. So you can see here about 26.1% of resistance for particularly beta-lactamase. All studies showed dominance of resistance for beta-lactamase, whatever is the class, and followed by multi-drug resistance and MLSP. And we also observed mobile genetic elements, which are very critical. They are responsible for horizontal gene transfer in environment. About 14 mobile genetic elements we have studied, and followed by aminoglycoside. You can see mobile genetic elements are more persistent during the analysis. So this is relative abundance of each of the thing with respect to heat map, and we also can get information regarding resistant mechanism, particularly what kind of mechanism prevailing with these things based on the data available. So here, in this case, antibiotic inactivation was the major mechanism, followed by antibiotic efflux. This was more or less same with all our other studies. So we also try to look into the microbial diversity to understand escape pathogens, because we are not directly culturing escape pathogen. Try to look into the diversity of the samples, and here also we got very good information, particularly related to what kind of prevalence those data. This is based on the phylum, class, order, family, genus distribution we try to present here, where Proteobacteria is dominant and Pseudomonas was the dominant strain observed in this particular study with respect to the community. So then we try to look into temporal variation during different periods, starting from December 2021 to April 22 data here we presented. So here also you can see the variation between the important class, how it is varying. In some seasons there is ingredient of multi-drug, in some season there is aminoglycoside. So we get data, particularly during the season of December to February, there is dominance of using certain kind of antibiotics that was detected. So this is the crude data in tabular form. From all the data points, about 49 antibiotic resistant genes are common in whatever period we have done. These are the common things. Among 123 detected, you can see in January and February there are more genes, antimicrobial genes we observe. So these are seasonal variation, how each of the gene is varying. So this kind of data we can, seasonal shift with respect to the different ARGs. So this we can do, and also I would like to say that we try to also compare with urban, semi-urban, rural communities, how these are varying with antibiotic resistance. So urban community for Hyderabad, semi-urban near to Hyderabad, 80 kilometers, where little bit of urbanization is there. Rural completely away from urbanization. So here interestingly 91 genes are common in 3 of the communities, but variation was significant. Urban community has a lot of resistance, basically with bacteriolactamase and MLSB, and rural communities only exhibited resistance to beta-lactamase. Semi-urban was moderate, but here because the rural area use less antibiotics, so resistance only observed for beta-lactamase. So these are the other versions how it is varying, that I will not go into deep, and also try to look into the mechanism, resistant mechanism with rural, semi and urban. So here we can see that the mechanism of efflux was more either to rural or urban, but inactivation is dominant in other two things. So this is the data, cross-sectional data, community, hospital, and industrial source. Community means again a area which we are studying for quite long time of period, and hospital, this is with respect to the project where madam is mentoring this project. So among 136, hospital showed less compared to the community because of a specific population. Industry area where pharma industry is there, we try to collect samples if they are releasing any antibiotics into the environment and have influence. So, but relatively, industry showed a less presence of antibiotic resistant genes, followed by hospital is more and community is more. So this hospital thing will help us to understand the infection, particularly that period patients are having. Based on this data, they can design the antibiotic which they are not resistant, particularly for a critical disease. This data will slowly help them to design that. So we try to analyze the data in different ways with respect to ARG classes. Particularly, our interest is to understand hospital, how it is varying, and gene-wise things also. And all are different. Particularly, industry has less gene prevalence. So these the data which we can analyze. Also looked into the resistant mechanism, how hospital, community, and industry is varying with respect to the mechanism. And each has its different mechanism. Particularly, industry is less infection, and wherever we see microbial, the gene transfer mechanism was prevailing. Whatever is the community, either hospital or a community or a industry. So then this is data from Mahakum. We try to look into the antibiotic because pseudospores play a major role in inactivating the infection rates in Mahakum. That's what many of the reports, and NEERI also has done in one of the projects. So we try to collect sample from Ganga upstream, Yamuna upstream, Triveni Sangam, and Yamuna downstream sites. And you can see that ARGs, MGs particularly, the variance was significant. Initially, before Triveni Sangam, there is a good amount of resistance and Yamuna sediments also. So, but it's varying. So, if we can dig deep into the data, we can understand, but we have to compare this data. Now, this month, we are collecting samples so that we can compare during Kumbh and after Kumbh, how this antibiotic resistance is varying. So these are the other which antibiotics it's more resistance in each of the site we try to understand based on the wastewater and sediment. So now coming to the inactivation, basically when AMR is coming into water infrastructure, that's wastewater infrastructure from domestic origin. So it has to be treated before releasing into the environment. So during this process, what is the fate of this ARG? We try to evaluate. So as you know, this is the common treatment system, and CDRI also has its own system, but this disinfection unit operation is important to break down the virus or bacteria which is having resistance things. So this has to be killed all bacteria to certain extent before entering into the environment. So we try to look into hospital also has a sewage treatment plant, and we try to understand how hospital inlet and outlet is working. So you can see the data. Simply, I would like to say among the 136 genes studied, so about 109 genes we detected in hospital inlet. Hospital outlet, about 87, still it is coming. So the proper disinfection is not happening because system was very small and not designed properly. So this is what we observed there, but we reported to them, but still they have to take action. So one of the treatment system, natural based treatment system, that's wetland treatment systems, constructed wetland, are very effective in reducing the microbial loads, but because of prevalence of aerobic and anaerobic conditions during the operation. So we have one system, wetland system, engineered wetland system, with 3 stages, Eichhornia, Hydrella, and Pista. The 3 stages treatment happen along with the bacteria with a COD load of about 340 mg per liter. So which is able to remove significant amount of COD. Here we try to see the fate of the antibiotics particularly. So you can see that there is significant reduction which each of the treatment unit, we call it as T1, T2 and 3T. 78% of the tested genes were detected in the inlet, and among this 62% reduction in first one and 757, finally 44% reduction was there in the final treated wastewater. So this is significant in the sense we are not using any chemical as a disinfectant. So again we can see which kind of thing is prevailing, which kind of antibiotic resistant genes are persisting based on the data. So another conventional sewage treatment plant, like normally what we used for all in all domestic purpose, this is again in ISCT about 14 KLD. Here also we studied with reference to the prevalence of these genes. This is the data, just a snapshot of the data I would like to take with respect to different antibiotics. Inlet, there are about 5 unit operations, and the reduction on a whole we can see there is reduction between 97% to 53% with respect to tributograph. Like that, each antibiotic we try to evaluate. But on the whole, particularly in January and February samples, we are able to detect more than 70% reduction of the antibiotic resistance gene with the unit operations. But each unit operations has its own mechanism of removing the antibiotic resistance. So this is other data like how it is varying those things. So here I would like to stop because of time. So what I means to say, this wastewater surveillance is one of the very important emerging mechanism which will play a major role in coming days. Particularly now WHO and other agencies also taking it as a mandatory to include in wastewater treatment systems. So 2 years back they have introduced in European Union to detect important these kind of virus and other things, selected things. So once in a week they collect sample and look into presence of these kind of virus, and AMR still not into not still considered, but virus and other things. So once they detect some kind of virus which has significant influence, they will know that something is going to happen. Then they will increase the frequency of sampling so that well before it infects the community, they will get an idea there is going to something outbreak of some kind of thing happen. So they will try to make early decisions and early management so that the epidemic will not go further. So apart from that, there are many other things which will help, which will come from these kind of studies. So a lot of scope is there, I would like to say. So I would like to acknowledge initially CSR for funding first project along with CCMB, and then DBT in INSAC project, which helped us to see the variants. And based on that mission mode project, where Madam is looking into that one. And also I'd like to thank our director ISCT. The whole of the work I have done in ISCT. That's still we are doing in ISCT, same project. And director CDRI for the project, which is AMR project at present running. Thank you very much for your attention. Any questions, ma'am? Oh, sorry.
Hello.
Next questions. Yeah, okay. I didn't see.
Thank you for this wonderful presentation. I was just wondering that when you detected presence of virus in the wastewater, did you see that the samples can reinfect back in, let's say, in a laboratory settings, something like that?
No.
Because as far as the viruses is concerned, they are found in the environment, but they're very fragile. So a lot of environmental factor might deactivate them.
Yeah.
And we have seen it for influenza virus. So like the sewage water, a lot of toxic materials are already there. pH is extreme. So under these conditions, the viruses may be found, but whether how many or what is the percentage of them remain active to infect back in the community, has that been checked, or you're planning to do something like that?
That part I am not sure, but whatever we have done, we try to collect sample, and we know that virus is very delicate. There will be breakdown of that structure, and that genetic material will be in the wastewater. And we also try to disinfect the sample by hypochlorite. We try to optimize. We don't want to get the live virus, so disinfect that virus, and then we try to compare with the live virus which is available in CCM. Standard graphs, all the things we have made with the live virus. Based on that, we try to understand how much load is there. Based on the two formulas are there at that point of time. After that, we have optimized. So that's why we didn't collect a single sample and showed that there is virus. But we collected number of samples from number of STPs, different conditions, and then we realized that this is virus which we are getting, but not in a live form. Most of the virus which we detected, the gene, even after hypochlorite addition also, there will not be live virus. So that material only we detected, and that is matching with the data. Particularly with Andhra Pradesh data, they try to give the reliable data, what their community is infected. When we compared from different cities, it is matching with that data.
So, thank you.
Yes.
Chetan has a question.
Hello, sir. Excellent talk. I am from Nagpur, and there are a lot of endemic viral diseases there, like Chandipura virus is there. Every year we report, a lot of NIV reports Chandipura, not a large outbreak, but small flare-ups. So apart from SARS-CoV-2, there are other very important viruses where we can detect from the fecal sample.
Yeah.
So are you planning, or do you have plan for like measles virus is there? Means it's we can get polio virus is there from fecal sample. So are you planning for other viruses as well?
So
a
actually,
surveillance program particularly, because it is an important indicator for larger outbreak, right?
Yes, so correct. So we try to look into other virus and other things also, but in ISCT, but I suddenly I shifted to NEERI. There we are trying. We have an epidemiology lab in NEERI, so we are trying to exclusively look out for Center for Epidemiology, trying to look for these kind of studies. These are very important. Polio was the first thing which detected in this kind of studies. WB in groundwater also.
Sir, I have a question. Whenever you were showing the data, which was for the antimicrobial resistance, the data related to the urban sector as well as rural sector, where you are getting 91 genes in common.
Yes.
So when you are collecting the samples, so in the rural sector, mainly the problem is random use of antibiotics in animals, especially for the poultry sector or for the cattle sector, where they randomly use antibiotics for treating the animals. So how would you differentiate that these samples that are coming from the rural sector are exclusively from the humans or from the animals also? Means, is there any possibility we can differentiate, because we are speaking about one health at a time?
Yes. So actually we have that idea, want to differentiate, but we didn't do that because of some thing, because we don't have the proper data what that rural area is looking into. But we attributed something to the agriculture chemicals which has been comes to runoff. That we have done, but we didn't see much of contamination coming into the water bodies, where we have collected with respect to the animal husbandry. But in one of the site in urban area in Hyderabad, we collected sample from site where lot of surrounding by lot of animal husbandry is there, but we didn't see that one.
So last question, Ashish.
Nice presentation, sir. So surveillance depends on the concept of incubation period, and most of the diseases, whatever we are discussing, have incubation period of 7 days to months. But suppose in future we have an epidemic having a very short incubation period of 1 day or 48 hours, something like that. So how we will plan for those epidemic surveillance?
So frequency of samples we have to increase in that case. Particularly this COVID, it has 30 days of incubation time. And what we try to do, we try to analyze sample within 24 hours. So of the sample collection. So there is a good window, 30 days window. AMR doesn't have any window, that depends on that sampling thing. So whatever we do, we try to do as soon as possible analysis so that any kind of detection will not lose. That's the thing right now I can tell. But this is a valid thing which needs to be looked into SOPs, particularly when we have that kind of virus. So in future, if any virus is coming, so yeah. That's we try to, like, it's a difficult thing because we have to get it to lab and do all the procedure, and near to 10 to 12 hours it required. In future, some kind of biosensors, electrocatalytic sensors are coming. If that works out, we can reduce the time.
So thank you for giving such an insightful talk, Dr. Mohan. These kind of studies of clinical environmental surveillance are very, very important for us to understand the upcoming epidemics, and also would be useful for public health decision making. And please give a round applause to Dr. Mohan. And I request the dignitaries to please come up on the dais. Please take. Yeah, please. So after the keynote lecture, we move on to the address by our chief guest, Dr. Shasany. But I would like to just give few points about Dr. Shasany, although Dr. Radha has already introduced him. Dr. Shasany's formal training is in agricultural sciences, which actually allows him to blend rural development with recent approaches in plant molecular biology. Actually, he started his scientific career in CSIR family. He joined CMAP, and then he went to ICAR lab as a director. This was National Institute of Plant Biotechnology, New Delhi. And he returned back to CSIR family to now serve as the director of NBRI. His research interests include engineering of medicinal and aromatic plants, bioprospection for human health. And through numerous outreach programs, his team has provided resources and training to empower the farmers of our country. Over to you, sir.
Thank you very much. First of all, Dr. Radha, thank you. This is an important subject, environment. Dr. Venkat, Dr. Kamal, and Dr. Ranbir Singh. So, as told by Dr. Venkat already, that's a nice initiative started in CDRI, and compliment to you. I mean, drug labs, starting the environmental initiative. But I'll just what Venkat told. Please find out the CDRI community antibiotic resistance gene also in some way. So that study can be utilized here. And already as I have, I don't know whether you will ask me questions. Before you ask me questions, then I will ask you questions. Please tell me the difference between plant and animal in one sentence. No. I say that plant they don't have locomotion and you have locomotion. So if there is any adverse atmosphere outside, then you run inside. If there is high heat outside, you will be liking to sit inside a AC room, but plant can't. They're standing there. They're standing there doing their own protection. And we are human beings much more intelligent, and we are learning from the plants and taking those things and trying to produce as drugs. I'm not telling you always you are producing drug from different sources, but plant is one of the main sources. 106 compounds are already in the allopathic system. So you know the importance of the plants. In later stage, if you say the antibiotics also, antibiotics are a small group, narrow group of antibiotics. So they are being repeatedly modified again and again to give us suppose analytic acid to that series of ciprofloxacin, norfloxacin, all this, and now that penicillin, beta-lactamase, now vancomycin has come, and again there is resistance for vancomycins. So everything is coming up. So now where do you go? We have to come to the plant system. But whether we are taking care of the plant. So anywhere you go, any places, then you find plants are neglected. But I am very happy to say that in CDRI campus it is green. I have seen Asoka plants, Michaelia champaka, and many other species, Kadamba. So there are many other species in the green campus of CDRI. Thank you for that. You are taking care. Again, if you ask, again I will ask a question so that you should not ask me questions. Tell me how much oxygen you intake every day. I'll tell. 0.75 kilogram. How much oxygen a plant gave, a full grown plant gave? Average 1.2 kilogram. So need a plant every day. So what is the average? If you see, average of India, the plants per person, around 28 plants, and it is reducing because we are increasing our population. We are increasing our population. When time will come, then 2% will share one plant. And do you know the average in Canada? It is around 9,000 plants. USA around 500. So we are 28 plants and reducing, gradually reducing. So wherever you go, I'm not going to give a scientific lecture, like wonderful lecture given by. This is one part of environment, antibiotics. Who has created antibiotics? We have. And if you say what is the environment, then soil, water, and air. The three things comes together. He is the director of NEERI. He will know much better about that, and he will tell much better. But we NBRI work on to some extent on environment. First thing, save the plants. Not that to get Padma Shri, you go and hold the plant like this. Save the plant in real means. Wherever you are there, you save the plants. That is a little bit modification in the lifestyle. So when I told about the oxygen and the plant scenario, and if we go on preparing development, going for making a road, you are killing trees. You are uprooting the soil. We are destroying the soil. Always it is happening. If there is some building is constructed, cut the trees. Fast things, cut the trees. Road making, cut the trees. Anywhere, development. So we are reducing in that way also. Again, climate change. The trees are, you can take drug. Now, how many people in this auditorium, they were not used to AC during their childhood? Can raise their hand? AC, air condition. But now you cannot sleep without air condition. I can sleep. No, you cannot. You cannot. Next day you will come to the institute and start behaving erratically if you don't sleep. So we need a better atmosphere. We need a better comfort. But to keep that comfort, what we have done? NBRI does the heat island studies. Heat island studies, as told by Dr. Radha. What this is, 10 years we are studying. We know where the heat islands are. We know which are the plants they can reduce the temperature. But who is going to implement it? I don't know. I don't have that much amount of money. I will go, that too in one place, in Lucknow. So there are many cities in India. So can you reduce the temperature by planting trees nearby that? And we know which are the plants giving more oxygen. So scientifically we know. If you talk about the trees, then the trees are bargat ka ped, people ka ped, neem, asoka, you have asoka, saraka asoka. These gives better oxygen in when they are full grown tree. And which are the herbs? Can we make a model, because we need that, which can precipitate the particulate matter because of the positive potential in the leaf, higher positive potential, and produce more oxygen? Can you put those plants in the, can you make model purifying the air? We use air purifier, which generate heat, which generate carbon dioxide. So anything in life you do, you generate carbon dioxide. Anything you do in your life, you destroy the plants. You use the plants. You destroy the soil. Not you, I also. Destroy the environment, air, everything we do. Because we look comfort. Preaching is very nice thing. But implementing in everyday life is very difficult. So always we know in any industry or any industrial activity, they should be accompanied by the green activity. There should be a green zone. How many industries follow? They are following. So this is policy matter. This policy matter, I will not enter into that. But when you see the soil, that is antibiotic scenario you have seen in the water, it is coming out. Soil, water pass through the soil, comes out of the soil. The sewage you produce comes to the soil. It goes to the soil, contaminates the soil. Another thing we are working on, arsenic contamination. There is heavy metal contamination. In rice, there are plants which take up the heavy metal very quickly. There are two things. If there is crop plant, it will take up the heavy metal and introduce into your grain and you will eat it, then you will die someday. The other plants, if you plant in the contaminated areas, they will take out these things and store there. You don't eat that. So there are sites where you can plant, go for tree plantation so that they can store the heavy metal and ameliorate the soil. They will take out the from the soil and put it in their body. So that can be used in different purposes, not for eating. So we work on arsenic amelioration. We work on petroleum amelioration, remediation. Again, sodic soil. NBRI is working in since long. So how you can make those soils which are already we have degraded? Because you might have listened, many area in Punjab and Haryana and Rajasthan, they have converted to sodic soil, acidic, alkaline soil. So gradually the crop area is decreasing, climate is changing, and when climate changes, because of our activity only, when the crop plants, their actually their period is changing, productivity. If there is no vegetative growth, there will be no production. Also, another thing is being found out that if the temperature increases, the climate changes, then the uptake of essential elements which we require, so those are reducing in the plant. Suppose iron and zinc, they are reducing. So they will come down to you, then you people will be more happy to develop drugs. So gradually different diseases are coming. These diseases, because of, you will say that I am connecting everything to plants. Yes, everything to plants. Plant can hold soil. Plant can conserve water. Plant can give you medicine. Plant can do oxygen, give you oxygen, everything. Let me come back to what we are doing. Microplastics. Recently, microplastics is a very phenomena which is nature paper has come recently that from the air how it enters into the body. It enters into the body from the air, from the soil, landfill sites. We know. And recently, the procedures and all these things, WHO, maybe WHO, World Health Organization, there is a meeting. Most probably NEERI must be there also, participate. So there we are producing a common protocol to detect. You detected. Then what? How you can take it out? So we are working on a protocol so that these are negatively charged. You make them negatively charged in the soil, nanoparticles. When you make them negatively charged, there should be one compound, plant compound, which is positively charged, which can aggregate them if you apply to the soil. Then again make some charge. I will not tell that which charge. Then take out, after aggregation, take out everything through some means. So we have to proceed in this direction to remediate. Already we have destroyed. And plastics, even if the rules regulation comes, still then we will put in our pocket and go. And very nice thing you have done today, that the COP has come. So you have removed the plastics, cups and everything. And you very well know that plastic cups or plastic bottles, if you keep something, then dioxins and everything being produced, that lead to cancer. And again, drug research institute will be very happy to produce drug for cancer. So let us cut the thing from the bottom so that less drug will be required. I'm not telling that you don't produce drugs. We should have drugs because it will never be ending scenario. Having said so, then again we have monitoring the monsoon. Means there are sinks in India. These sinks actually call the monsoons. You say that every year monsoon comes. But there are some reservoirs, some biosphere reserves, which actually create the condition in India so that monsoons is being called. Because the biosphere resources are shrinking, for that the monsoon is erratic. So there is a correlation. So we monitor in different biosphere, at least 2 biosphere reserve. In Meghalaya and in one Odisha, that's the biggest biosphere reserve in Similipal. It has been termed that the monsoon, it calls the monsoon. So how the plants, they are actually calling the precipitation. How they are actually, this is creating an atmosphere of carbon dioxide or oxygen. What is there in the soil, microbe, how they are doing their activity, what type of gases they are releasing. So all these things, then in future maybe your biosphere region may be shrinking, so you can have a model to call the monsoon artificially, because everything now we are going artificially. So everyday life. So you start in the morning, go for the toothbrush. Toothpaste, so like aroma. The aroma is carbon. This is from the mint plant. You go for a sabun or anything, so you like aroma every day. So all the aroma are being inspired by plants. Even if the synthetic aroma, unless and until you understand the plant aroma, you cannot understand the synthetic aroma. So you require everything in your life from the plants, and all the industries, suppose rice, Pepsi, any industry, they are using plant product. So if you go on destroying the environment, how we are going to keep our crop plant? So we need to think right now. So there are many things we work on. I have 15 minutes. So I will stop here, because after listening a wonderful lecture on antibiotics, I am not going to speak more about any other thing. But wonderful that your campus is very nice, and you have started 3 projects which are actually beneficial for your health and for the environment. And I must give compliments to you as a team, CDRI, and many more things. And my thoughts are synchronized with Radha. And the last sentence she told when she was speaking, always I synchronize with that sentence. Maybe you can take a pledge in future to change our lifestyle and help the plants to grow and keep the plants and conserve the plants. Another thing I will tell you before leaving, after 1 month or so, please visit the campus in Banthara. So there we have conserved the germplasm plants, we have conserved the bamboo germplasm, we have developed a heritage garden. This heritage garden is the garden which you have inherited. This has been cut. Means like I'll give you example. बाबा निमली आप लोग जानते हो ना, बाबा निमली, ऐसे ही अक्षय वट आप जानते हैं। ऐसे हमारा हेरिटेज प्लांट जो है, द फर्स्ट वार ऑफ इंडिपेंडेंस, जिसमें लोगों को लटका गया था, फर्स्ट वार ऑफ इंडिपेंडेंस, चंपारण रिवॉल्यूशन, दांडी मार्च, जहाँ जहाँ महात्मा गांधी जी ने बैठे थे, नमक बनाए थे, वो सारे प्लांट्स का ओरिजिनल कॉपी from the original plants, cut and rooted and planted with QR code. You can go and listen to the history. Also the plants in the cellular gel, which has witnessed everything in the cellular gel. The plants in Jallianwala Bagh. So Nursery is complete. Now we have collected from Tamil Nadu the heritage plants. The track is ready, so we will be planting them. So all the plants, maybe by mistake somebody may cut it. So we should have the replica. NBRI should conserve those plants, the heritage plants, so that you can learn from our past. Then come back in the track. I'll have if you cannot walk, I have a e-vehicle. You can sit there and move around. Then come to a 4 replicates. We have maintained the trees of Uttar Pradesh in no Gangetic plain. 4 replicates. This is jungle. Then go to the natural jungle. There we have conserved natural jungle. Then Miyawaki forest. Then see the endangered plant enclave, that is in a shape of the India map, where the plants are endangered, in that place they are being planted. Then come to, after seeing that, then go to the Nakshatra garden and Navagraha garden. The plants which you use, actually think about our Nakshatra and Navagraha. Then come and take a cup of tea and come back and relax and come, go home. Then maybe before I make it 100 rupees ticket, you can visit that after 2 months. Right now not. It is under making. So thank you very much again for calling me here. I am glad that you are listening to me. Many people from the backside, they run away. They thought, what, this is not related to drug, and that side, they're gone. I am marking this thing, how many people they have gone, where they are sitting there. So thank you very much. Jai Hind, Jai Bharat. Namaskar.
Thank you, sir, for sharing your thoughts and also highlighting, mindful activities that we can do at individual level to conserve the plants. वनस्पति जीवन है, and चलो इस जीवन को और सुंदर बनाएं through our, sustainable practices. Thank you again, and I request Director to felicitate both the guests, first Professor Mohan and then Professor Shasany. So, on the behalf of CDRI family, this is a small token of appreciation. Yes. Yeah. I request Mr. Jain to present the vote of thanks.
Good afternoon, everyone. On behalf of CSIR-CDRI, it is my privilege to extend a heartfelt vote of thanks on this momentous occasion. The inauguration of our 1.2 megawatt rooftop solar power plant and bio-composter, a significant stride in our commitment to sustainable and green science. First and foremost, I extend deepest gratitude to our chief guest, Dr. Ajit K. Shasany, Director CSIR-NBRI, for gracing the event with his presence and sharing his valuable insights. A special thanks to our guest of honor, Dr. S. Venkata Mohan, Director CSIR-NEERI, for joining us today and inspiring us with his visionary thoughts. We are equally grateful to Dr. Radaha Rangarajan, our Director, for presiding over the ceremony. Her legendary leadership and unwavering commitment to sustainability have been instrumental in realizing this initiative. I also wish to thank all our colleagues, collaborators, and staff whose efforts and dedication made today possible. Thank you so much.
Thank you all. We close this program.
Thank you.